Welcome to our second episode of Know Your Supplier. My name is Brian Mperi. I'm the Managing Director for Celestial BioSystems, an authorized dealer and authorized agent for Innovative Biotech in Zimbabwe. Today we are going to look at sample collection and nucleic acid extraction solutions from Innovative Biotech. So on sample collection, we have solutions for life sciences and molecular diagnostics. So we are going to divide these into two. The first will be life sciences, and then the second will be molecular diagnostics. And on life sciences, this is where you get the majority of all the lab work, whether it's research, whether it's uh, academics or any other, you know, general laboratory workflows. Those can be classified as life sciences. And then I've got molecular diagnostics, where we are dealing with mainly clinical samples for the purpose of patient management, surveillance, or demographic studies, and so forth. So, so we're going to start with life sciences. So on life sciences, we have got sample collection solutions, mainly from Zymo Research, and the main component or the core reagent of all these sample collection devices is our DNA RNA shield, which is a stabilizer that would last for more than 2 years at room temperature for DNA and more than a month at room temperature for RNA, and it is suitable for different sample types. That includes saliva, blood, stools, and that's also your plant samples, your environmental samples. And then we have got self-collect saliva collection kit, which is a user-friendly and a spill-resistant saliva collection device that you can use from home or in the field. And then we have got the DNA RNA shield swab collection kit. This comes in two forms or with two components, with a swab and a DNA RNA shield in another container. So this is pre-filled tubes with sterile swabs for collecting from the throat, nose, and the buccal region. And we also have the fecal collection tubes. So these are pre-filled with reagents and a scoop for efficient sample collection of fecal matter. Um, so basically, you would be able to collect about 1 gram of fecal matter with this collection device. And then you have got bunny wipe. Bunny wipes are the non-invasive collection tool that amplifies tool collection, that simplifies, simplifies stool collection, designed for easy use in home settings. Also, the bind wipes are those that you actually stick on top of your chambers, and yeah, and you can actually efficiently collect fecal matter using the bind wipes. And then you have got the urine conditioning buffer. So these are used for stabilizing nucleic acid in urine samples, and I'm pretty sure most of you would know that urine samples have got quite a number of uh, should I say maybe contaminants or inhibitors that will definitely interfere with your PCR and nucleic acid extraction process. So you would definitely need a buffer for that to make sure that you stabilize your nucleic acid within those samples so that you don't degenerate your DNA or your RNA. And then you've got blood collection tubes. So these are specifically designed for stable collection and transport of whole blood. And even within these blood collection tubes, you still have your DNA RNA shield reagent. So in most of these, if not all of them, the main component, like I said initially, it is your DNA RNA shield. your stabilizer. So this is the core reagent within all the collection devices that we have, and then they are subdivided into different applications or different settings under which you would want to collect your samples. So like I mentioned, the main brand here would be Zymo Research, and from Zymo Research, that is where you get your different sample collection devices for different applications. So if you go to the Zymo Research website, it is one of the websites that is very easy to use and follow, and it can actually help you to get the correct product for the correct application. So there are filters. When you're dealing with different sample types. So you've got fecal matter, for example, blood, swab-based samples, saliva, urine. And then on the filters, you can actually select the different sample types that you're dealing with. Are you dealing with tissue? Are you dealing with urine? Are you dealing with vaginal samples? Are you dealing with wounds? Dealing with water? Dealing with environmental samples? And so forth. There are quite a number of different sample types that you would find on that drop down, and you have got also different features and formats under which you would find the sample collection and preservation devices. So you can also try it out. Check on the Zymo Research website on how you can actually pinpoint a certain collection device from the website for the particular sample type that you're dealing with, and you'll see how convenient, efficient it is. And then the second thing is molecular diagnostics. And our main brand for molecular diagnostics, which I'll focus mostly in here, will be Seegin. So from Seegin, we have got sample collection devices and pre-treatment solutions. Um, so we have got various sample collection devices also for molecular diagnostics applications that support uh, different sample types, but mostly I'm sure you would know that with molecular diagnostics you'll be dealing with respiratory infections, respiratory viruses. You will dealing with um, pathogens like human papillomavirus, HPVs. You'll be dealing with STIs. You're dealing with you know TB. You'll be dealing with gastrointestinal infections. So you would need sample collection devices that actually support a wide range of sample types. So in here, what I've listed, we have got the nasopharyngeal or oropharyngeal swabs, which are the main uh, uh, products that are used across different applications of different pathogens or syndromes that you'd want to detect using molecular diagnostics. And of course, you've got saliva, you've got sputum, you've got urine, you've got blood, you've got stools, and then you've got the bronchoalveolar lavage, the BAL fluids. Yes, and then you have got with Seegin, you have got validated VTMs that are compatible with the various platforms that we have for nucleic acid extraction and uh, and uh, and Seegin. So we do have various compatible devices that can work with Seegin nucleic acid extraction platforms. And then I'll also jump and go to extraction-free methods. So we also have got assays from Seegin. We have got selected assays that actually allows you to do a direct uh, that will allow you to do direct sample processing without really doing nucleic acid extraction. So you would need a special collection device for that. Usually these are devices that will actually lyse your sample, your cells, and release your nucleic acid from the cells so that they can be free floating within your media or yeah, within your media that you would have used for collection. So we have got the Allplex SARS-CoV-2 fast PCR. We have got uh, the Allplex SARS-CoV-2, flu A, flu B, RSV. I mean, we've got quite a number from Seegin that actually allows you to do direct sample processing from the primary sample after having collected with a certain collection device, and you can use that as your primary sample in PCR directly without you having to do nucleic acid extraction. But they are, pre-treatment solutions that are specified or validated to be used for that, for extraction free methods. Uh, and then of course you've got stabilized controls, which uses these are molecular controls that mimic whole pathogens and are inactivated to meet laboratory safety requirements, like your IC, your internal controls. Yeah. So that's it on molecular diagnosis. I mean, if you have got any questions, you can also reach out so that we can look at that inquiry in detail. So from Seegin, the most prominent device that we use for sample collection will be the inert swabs. So the inert swabs actually apply across various sample types, so across various applications, across various pathogens. So it is one of the main sample collection devices that we use for molecular diagnostics. So these will be the inert swabs. I mean, for respiratory viruses, you would need a swab. For STIs, I mean, you would need the swabs like vaginal swabs and so forth. For GIs, the gastrointestinals, you'd also need swabs. Um, so you see, it is a device that can actually use across different applications and different pathogens. And jumping from sample collection devices, we will go straight into nucleic acid extraction solutions. So we have both manual and automated nucleic acid extraction solutions. And like I also mentioned in the first episode, that the majority of our nucleic acid extraction solutions are from Zymo Research. Um, and we have got automated nucleic acid extraction solutions. I mean, forget my typo there, under automated. So I wrote like nucleic, but it's nucleic acid extraction solutions. So we have got Seegin for molecular diagnostics, and we have Eppendorf for life sciences. But you can actually customize your EP motions from Eppendorf, whether to do life sciences, nucleic acid extraction, or molecular diagnostics related nucleic acid extractions. So we do have got both. So we will look a bit into nucleic acid extraction solutions from the manual side. So we still go back to Zymo Research, and what I like about Zymo Research is that it has got quite a number of nucleic acid extraction solutions. And um, these nucleic acid extraction solutions are validated for specific sample types. So it's not a one size fits all. I'm sure you would know that different samples are made up of different material, different exoskeletons, different membrane materials. So you would definitely need different nucleic acid extraction solutions optimized for that particular sample type for you to get optimal DNA, optimal yield from that particular sample. So it is very critical that you select the correct nucleic acid extraction solution from our Zymo Research pool of nucleic acid extraction solutions. So on manual here, when you go to the Zymo Research website, you can actually go to products and go to DNA and RNA extraction solutions. So you will find here that you have got DNA extraction solutions for biological fluids, cells, and tissues. You will have for microbiomes or environmental samples. And let's say, for example, you're doing whole genome sequencing, which you can also do with us. You have got high molecular weight DNA. It is very important when you're doing long read DNA sequencing that you get high molecular weight DNA extracted. So you'd want to use an assay or a kit that is not optimized for high molecular weight DNA extraction when you want to do next generation sequencing. And then, of course, we have got cell-free DNA, and then we have nucleosomal DNA. Of course, we have got plasmid DNA and the different plasmid DNA samples. And we also have got the DNA cleanup and concentration kits. And one of the mostly popular products will be your gel DNA extraction and the clean and concentration kit. Your gel DNA extraction, whereby after you're running a gel, let's say it was perhaps a nested PCR, and then you are interested in particular band, you would definitely go on to do gel DNA extraction. So you wouldn't want to use a different kit for extracting DNA from a gel. Like I mentioned, I mean, all samples are optimized for a particular sample. All sample extract, I mean, all samples have got different makeup, and all DNA extraction kits are optimized, to extract efficiently from a particular sample type. So it is very important that you select a, a correct or an ideal kit for a particular sample type that you're dealing with. And of course, we do have or total nucleic acid extraction solutions where you can actually co-purify your DNA and your RNA, and that also depend on the sample type. And of course, they are automated kits that you can adopt from Zymo Research using different automated platforms. Um, so these are your high throughput solutions and automation. So we do have got the same kits that you can actually use with your EP motion, for example, from Zymo Research or other platforms that you may have in your lab. So you can also get in touch on this, and then we can look at a particular inquiry in detail and according to how you want to to proceed with your nucleic acid extraction um, solutions for manual DNA extraction or RNA extraction. And then you have got automated solutions, um, both for life sciences, molecular diagnostics. So we have got our EP motions, like I mentioned, and on molecular diagnostics, if you're running Sagen assays or you're interested in running Sagen assays, we actually have the Starlet EOS, so which is our sample in, result out system, where you can actually even feed in into the platform primary collection uh, samples, primary collection sample tubes, like if it is blood, you can have your blood collection tubes. If it is your 1.5 ml tubes, you can actually put that in. If it is your 2 ml tubes, yeah, the primary tube that you use for collection, you can actually put those into the platform. There are different configurations that would allow you to actually put in those various primary collection devices into the machine. It would efficiently do your nucleic acid extraction, PCR, result, interpretation, and so forth. So that's your started AOS for molecular diagnostics. So without wasting much time, that's it for today, and you know where to find us, and you know why you would always choose Celestial BioSystems for your laboratory solutions. Thank you very much.